Hello everyone and welcome to our presentation. It will be based on strategic control of select parasitic infections. The presenters are Delia Dudnot, myself Veer Farhad, Tiffany Prince, Callista Barnard, Maria De Souza, Mike Fordyce, Jonica Williams, and Miracle Morgan. This presentation is designed to give you a comprehensive overview of current and emerging strategies in malaria, giardias, and leishmaniasis. So let me start off by saying what are parasitic infections. Disease known as parasitic infections are brought on by creatures called parasites that reside inside or on top of their host and obtain nutrients at the expense of the host. Millions of people worldwide suffer from parasitic infections, which are particularly common in tropical and subtropical areas where proper sanitation and hygiene may be lacking. Now, why is control of parasitic infection important? For a number of reasons, parasitic infection control is essential. Now, the first one here is impact on public health. So this is saying from minor gastrointestinal issues to serious life-threatening illness like malaria. Parasitic infections can result in a variety of health issues and even death. The second one here is economic and social burden. Now, because of medical expenses, lost productivity, and permanent disability, these infections may result in large financial losses. And lastly, preventing transmission and consequences. If treatment is not received, a number of parasite infections can result in fatalities or long-term consequences.
Building on what we've discussed, here are key parasite control strategies in three areas: vector control, hygiene, and medical implementation. In vector control, insecticide treated bed nets cut malaria deaths by 20% overall and 60% in young children. Indoor residual spraying reduced transmission by up to 80%, while managing mosquito breeding sites can lower mosquito density by 90%. For hygiene, regular hand washing cuts parasite transmission by 30%. Improved latrines reduce helminth infections by 45%, and wastewater treatment removes 99.9% of parasite eggs, protecting crops. Finally, in medical implementation, mass drug administration lowers lymphatic filariasis by over 70%. Integrating care into primary health boosts coverage by up to 60%, and regular deworming reduces infection intensity by 70% in school children. Together, these strategies offer a strong, coordinated approach to parasite control.
I will now be covering malaria causes and control strategies. Firstly, malaria is a parasitic infection done by the Plasmodium species and can be found in areas that are warm and humid, for example, Caribbean countries such as Guyana. The Plasmodium species is transmitted via the female Anopheles mosquitoes, which acts as an intermediate by biting humans for the parasite to get to the bloodstream. Humans can be infected in another form, which is by contaminated needles and from undergoing blood transfusions. For the control strategies, diagnostic measures can be taken, such as analyzing blood smears with a microscope to detect the parasite causing the infection. And lastly, through vector management by implementing the use of insecticides and bed nets to decrease the rate of the malaria spreading, leading to less mosquitoes populations over time.
I will now be looking at the challenges and improvements for malarial control strategies. Challenge 1. Anti-malarial drug resistance. Plasmodium falciparum has become resistant to almost all commonly used anti-malarial drugs, example chloroquine. This has been amplified by misuse of anti-malarial drugs, utilization of widespread substandard or counterfeit drugs, and weakening of some drugs by long half-life, increasing parasitic adaptation. Improvement. Artemisinin-based combination therapies. ACT has become the standardized treatment highly effective against Plasmodium falciparum and WHO has terminated all monotherapies by pharmaceutical companies in January of 2006. Challenge 2. Inadequate diagnosis. The rate of accurately confirmed malaria diagnosis is often low and results in an overuse of anti-malarial drugs, contributing to parasitic drug resistance. Improvement. Introduction of rapid diagnostic tests. RDTs has made diagnosis easier, faster, and accurate in improving confirmation of malaria before treatment in patients, reducing the misuse of anti-malarial drugs.
Moving on to Giardia. Giardia is a microscopic protozoan parasite that causes giardiasis, a diarrheal illness in humans and animals. The species affecting humans is Giardia lamblia, aka G. intestinalis or G. duodenalis. It exists in two forms: the cyst stage, which is inactive but resistant and survives outside the host, and the trophozoite stage, active and disease-causing, living in the intestines. Some causes of infection are drinking contaminated water, example water from lakes, rivers, and pools, eating unclean food or food prepared by infected individuals, person-to-person contact, especially in childcare settings, and contact with infected animals like dogs and beavers. Some control strategies are water treatment. Ensure to boil water, use proper filters, or disinfect with iodine or chlorine dioxide. Personal hygiene. Wash hands properly, especially after restroom use or handling animals. Food safety. Rinse produce with clean water. Cook food safely and avoid food from sick handlers. Avoid unsafe water. Don't drink untreated water from natural sources. And finally, clean environments. Regularly disinfect homes and childcare areas and ensure proper sewage systems.
I will now be looking at the challenges and improvements for giardiasis control strategies. Challenge 1. Drug resistance. Both assemblages A and B of giardia show increasing resistance against first-line drugs like metronidazole and tenidazole, particularly in patients returning from endemic areas like South Asia. Some strains display multi-drug resistance, reducing effectiveness of alternative treatments like albendazole. Advancement in treatment strategies. Effective treatment strategies have been adapted to address drug resistance, including combination therapies with combining medications like albendazole and nitropidazole, and alternative medications like quinacrine, which have been identified as a potential treatment for nitramidazole-resistant giardiasis. Challenge 2. Limited diagnosis. While there are several diagnostic tests for giardiasis, such as microscopic, immunological, and molecular techniques, they are all limited and it is difficult to identify the best procedure for diagnosis. While microscopy remains the standard due to accessibility, it is notably less sensitive for diagnosis than procedures like PCR, which is underutilized due to cost and infrastructure constraints. Furthermore, there is a lack of point-of-care diagnostics for resistant pathogens, which hinders timely treatment. Improvement, enhanced diagnostic techniques. Development of techniques like polymerase chain reaction, PCR, enzyme-linked immunosorbent assays, ELISA, and direct fluorescent antibody testing offer high sensitivity, enabling the detection of giardia DNA for faster and more accurate diagnosis.
Now, let's take a look at leishmaniasis. You might be wondering, what is leishmania and how is it caused? Well, let's waste no time and get to it. The infected female phlebotomine sandflies bite people, exposing them to protozoan parasites of the genus Leishmania that causes leishmaniasis. Transmission mode usually takes place in tropical and subtropical areas, and note, there are over 20 species of Leishmania that are known to cause disease in humans. The parasites infiltrate and proliferate within the host macrophages, resulting in a variety of clinical presentations, including cutaneous, mucocutaneous, or visceral leishmaniasis. Now, let's look at a control strategy which includes vector management. To avoid getting bitten by sandflies, use bed netting sprayed with pesticide and control of the environment. Early detection and treatment of affected person can lessen the severity and spread of the disease. To break the cycle of transmission, diseased animals, especially canines, should be treated urgently. Education of the community about illness symptoms and prevention strategies, and we need continuous work to create vaccines that can work. With that being said, let's move on to the challenges and improvement of leishmania. Control 1. Leishmania spread through a variety of species and reservoir, making it very hard to control. Improvement 1. According to the WHO 2023, the use of treated insecticide bed nets and by removing infected animals have shown to be very effective. Control 2. Despite numerous researchers to date, there is no vaccine for leishmania, and due to its complex life cycle, endemic populations remains vulnerable. Improvement 2. Experimental vaccines with a combination of recombinant antigens, live attenuated strains, along with DNA, and with significant advances in immunology and molecular biology, we anticipate and hope for a future breakthrough. In concluding, parasitic diseases like malaria, giardia, and leishmaniasis continue to pose significant health threats globally, especially in underserved regions. While advancement in diagnostic treatment, hygiene, and vector control have improved disease management, challenges such as drug resistance and diagnostic limitations hinder complete eradication. Thus, sustained research, innovation, and collaboration remain crucial for reducing the burden of these infections and protecting vulnerable populations. These are our list of references, and thank you.